All righty, let's go ahead and get rolling. We have reached December. We have to finish up the autonomic nervous system today. And then that will leave us just a little bit of sensory stuff for Monday. With Monday being right before the exam, it will not be a heavy lecture. More just kind of reviewing a lot of the stuff we've already kind of talked about, putting it together in the sensory systems, and then also we'll have plenty of time for review for both the lab and lecture exam and for the final exam. So come with your questions for that. Also on Monday, you have your last two assignments, your final unit review, unit 14, and then also a fun reflexes lab where you're doing procedure 2 and only procedure 2 on pages 371 through 374, and then also procedure 2 through 380. Again, do what you can of those. Any activity you're not able to complete, please answer it as a thought experiment, right? What you think would happen if you're not able to do it. And then it's all exams. You have your lab and lecture exam on the 8th, and then you have a full week to prepare for your 100-point cumulative final exam. Again, that's 100 multiple choice questions. You'll have 2 hours to complete it, just like a normal final in the classroom, and then you are done. Then you can do your Christmas shopping. All right, questions on any of that? All righty then. We left off last class and we had completed basically the left side of this scary octopus looking illustration where we'd done the parasympathetic nervous system. And so now what we need to do is we need to look at the right side of this and talk about the sympathetic branch of this. Now, again, as we've talked about, the sympathetic nervous system is very elaborately branched. So while we're going to talk specific pathways, remember there's going to be lots of collateral pathways that will allow this as well. So we're primarily going to be talking about the general trends of how this works, and know that there, of course, are going to always be exceptions to something like that. Hopefully, one of the first things that jumps out to you when you look at the sympathetic version of this, and here we see another nice illustration just emphasizing it, one of the big differences between the sympathetic and parasympathetic anatomically is that, hold on 1 second. Okay, is that where our preganglionic neurons are located. Notice that unlike the parasympathetic, where some of them were up here on the brainstem and some of them are down here in the lower part of the spinal cord, which is why it got the name craniosacral from an anatomical standpoint. Notice here, all of the preganglionic neurons in our sympathetic nervous system are all located in the lateral gray horn of the spinal cord between T1, oops, let's change colors, between segments T1 and L2. So not surprisingly, anatomically, we would refer to the sympathetic nervous system as the thoracolumbar because it's contained within the thoracic and the lumbar region of the spinal cord. So obviously one of the big differences is that starting point in the central nervous system. And so not also surprisingly, are there differences in the structures that are formed by their axons. The other big difference is the ganglia as well. Well, there are many big differences, but another big difference between these is in the ganglia as well. Notice, let's start here. There are primarily 2 types of ganglia. There are these representing the sympathetic chain ganglia, and then we have these over here representing what are known as the collateral ganglia. We've actually seen these before, partially anyway. If you remember this picture when we were focusing on the spinal cord and we were looking at the 4 branches, our dorsal root and our ventral root, remember, come together to form the spinal nerve. And then we had our ventral ramus, pardon me, our dorsal ramus, our ventral ramus. And remember, we had the 2 rami communicantes, the white and the gray rami. And remember, we said they went to these sympathetic ganglia. Notice these sympathetic ganglia are interconnected with each other, which is why they are sometimes referred to as the chain ganglia. These are also known as the trunk ganglia. And notice they're paired. There is another set on the other side. We can't see all of them, but we know that they're going to go down the other side of the vertebral column as well. So these chain ganglia are paired, meaning that there are two of them, one on the left, one on the right, and they run parallel alongside of the spinal cord. So these are also referred to as the paravertebral ganglia. Now, what this illustration doesn't show, and I'm going to cheat and erase the line that I just drew there, is that notice there are additional axons that are coming off towards the front. And a little bit lower here on the spinal cord, they will connect with another set of ganglia that are located in the front of the spinal cord. These additional ganglia are what are known as collateral, because they're to the side of the main pathway. They're located on the midline, so they're unpaired. They're just a single ganglia, and in fact, there are only 3 of them. And because they are in front of the spinal cord, they are also known as the pre-vertebral ganglia. So we have 2 sets of ganglia. We have what we can call the chain ganglia and the collateral ganglia, or you can also use the terms paravertebral and prevertebral. Both are acceptable ways of describing these ganglia. And not surprisingly, they are located in different regions of the body, different regions in association to the spinal cord, so they are going to both innervate different parts of the body. Our paravertebral ganglia are going to be the ones that go to the organs above the diaphragm and to all of the skin. Whereas the collateral ganglia go to the organs below the diaphragm. So not only are we going to see a difference of them anatomically, we're going to see a difference in their pathways and a difference in their functions as well. Let's go back to one of our octopus drawings here, where we can see some of this. And again, as I mentioned, we have this row of chain ganglia that are all interconnected to each other. They are named based on their associated region with the spinal cord. So obviously some of them are closest to the cervical region, some to the thoracic, some to the lumbar, some to the sacral, and we have the final single coccygeal chain ganglion. However, these vary, the number of them. The number of them varies from person to person. Almost everyone has 3 cervical ganglia. Because we have 3 cervical ganglia, we can easily label them and identify them, and notice they indeed do that. The superior cervical ganglion, the middle cervical ganglion, and the inferior cervical ganglion. Notice I did say almost everyone, For some individuals, the middle and the cervical will fuse together into a single ganglion. But as we see, they both innervate the same structure. So whether it's two structures or whether it's one structure really doesn't matter. Also, every single person has one coccygeal ganglion. But the numbers of thoracic, lumbar, and sacral, all of these vary. So if, for instance, I were to point an arrow at this ganglion right here, do we know for certain whether that is thoracic 10 or whether that is lumbar 1?
No.
No, of course not. So we're not going to have to worry about identifying any of the ones in between, right? I could certainly point at one of the first three, and you would need to be able to identify it. I could certainly point at the bottom one, and you need to be able to identify it, because like I said, those are pretty consistent. But all the ones in between, you don't have to worry about identifying by name. Just know that they are chain ganglia. Now, these three cervical are very special with very specific functions. But our coccygeal is special as well. And I need another picture to show you why. Notice here, remember our chain ganglia are paired, meaning that there is a left side and a right side. So notice there is a left superior cervical and there is a right. And again, if I'm thinking anatomical position, I did that backwards. This over here would be the right and this over here would be the left. But there's, a cervical, superior cervical chain ganglia on the right and one on the left. And they run parallel to each other, parallel to the spinal cord, which is why we call them paravertebral. But notice what happens at the very bottom of our chain ganglia. The two coccygeal ganglia actually fuse together into a single structure. So instead of two truly parallel lines, the inferior part of the chain ganglia actually come together, forming this big, large U shape. And as we know, our anatomists love to name everything. So these two fused coccygeal ganglia at the bottom of our chain are what are known as the ganglion impar. So it's just a fancy name for those two coccygeal ganglia that fuse together. And don't ask me why they fuse together. I don't have a good answer for you as to why that occurs. It just does. So it's just the way that it is. All right. Questions on the anatomy of this? All right. Now, notice as we look at this illustration, we can also see some of the functions. Now, in general, as I mentioned, this innervates all the organs above the diaphragm and the skin. But let's be more specific. Let's look up here. Oops, sorry, I don't have my chat window open. Oh, okay, just a response for me. Perfect, excellent. Let's start at the top with those three cervical chain ganglia. Notice the superior cervical chain ganglia basically provides all sympathetic function of the head, both external and internal. So salivary glands, mucus production in our nose, tear production, right? Sweating, hair standing up on end, any of those functions, dilating blood vessels, right? When you're embarrassed, all of those things, any sympathetic function of the head comes through this superior cervical chain ganglion. All right. Think of how this compares to our parasympathetic. In our parasympathetic, we had one ganglion that innervated the tears. We had one ganglion that innervated, two that innervate the salivary glands, right? All of it was much more discrete, much more local, whereas this big global effect. The superior cervical chain ganglion basically does anything in the head. And again, if you cheat and look at the illustration, you'll say it even bleeds a little bit down where it can affect the heart as well. That heart input is minor, but it's still there. Because again, big, broad effects. The next two, the middle and inferior cervical chain ganglia, which for most people are separate, but like I said, in a few people will fuse together. Notice their functions are essentially the same. They innervate the organs of the thoracic cavity, primarily the heart and the lungs. Any internal organ, not in the head, but above the diaphragm. So primarily the heart and lungs, but they can also affect some of the blood vessels in there. And that's pretty much everything that's in the thoracic cavity. That leaves a whole lot of chain ganglia left, doesn't it? Well, and it turns out all of the remaining chain ganglia basically all have the exact same function. Every other chain ganglion from T1 all the way down to that coccygeal all innervate the skin. Anything associated with the skin. Sweat glands, right? Sudoriferous glands, erector pili muscles, right? Blood vessels and adipose of the skin. And again, as we mentioned, it does vary from person to person. Everybody has somewhere between 9 and 12 thoracic, 4 to 5 lumbar, 4 to 5 sacral, and of course that one coccygeal. And these are responsible using those dermatomes. Again, it's not random. Using the dermatomes, they're going to control that skin in a specific region. These form part of the spinal nerves. Remember, as we talked about, if we cut the nerve to the finger, we can't feel with the finger, we can't move the finger, and because we've cut this, we won't be able to make our erector pili muscles stand up, we can't sweat from the finger, we can't dilate the blood vessels from the finger, we lose sensory motor and sympathetic function to the skin, all by cutting that spinal nerve that goes there, because that's their route that they're going to take. So again, we have almost 25, somewhere around 25 chain ganglia, but really just those 3 major places they can go: head, thoracic cavity, or skin. All right. Questions on that? Again, here we see this nice superior view. This is a view we've seen before when we were talking about the spinal cord, when we were talking about the protective coverings and everything else. But again, here we see that spinal nerve forming our dorsal ramus, forming our ventral ramus, forming the 2 rami communicantes, right? Remember, it is the white one that is lateral and the gray one that is medial. And they go to those chain ganglia that are located just along the side of the spinal cord. And in fact, if you get the opportunity, hold on. to go to the classroom, I don't know who, if anybody, has had the chance to go to the classroom, but if you get the chance to go into the classroom, we have this model in the classroom, which is affectionately known as the baby back ribs. It is a plaque, three-dimensional plaque that has the ribs and stuff on it, which is why we call it the baby back ribs. And I don't know how well this shows up small on your screen, but if you get the opportunity to go, what you will notice is it shows the spinal nerves coming out. It shows the plexus, the cervical, the brachial, the lumbar, and the sacral plexus. So we can see those all really, really nicely. But if you get a chance to look closely at it, the other thing that you will see is right here, we have our 3 cervical chain ganglia. We have the rest of the chain ganglia that work their way down, all the way down to the bottom and then start to come together. And when you look really closely, you can actually see the gray and the white communicantes that are coming off of it. Notice this also does a really beautiful job of showing the terminal part of the spinal cord, the conus medullaris and the cauda equina coming out of this down here. So this is a really, really nice model if you get the opportunity to go in the classroom and take a look at it to see some of these features that we are being held responsible for. All right, back to this. And again, notice now we come back to this, and again, it is the white ramus, which is the lateral ramus. It is the gray ramus, which is the medial ramus. And again, they both go to the chain ganglia. And notice each chain ganglia has a white and a gray ramus that comes into it off of each segment of the spinal cord. All right. Questions on that? All right, perfect. From there, we can talk about the 3 collateral ganglia. Now, again, this is the one part, I love these pictures a lot, but this is the one part about it that I don't like. Again, these are called collateral ganglia. And when we think of the term collateral, collateral does mean to the side. And in the illustrations, they always show them to the side. But are these ganglia really way off to the side of the spinal cord?
No.
No, remember, these are located right here, unpaired, right here in the front. So we have these 3 right here in the center, which is why they are known as the pre-vertebral ganglia. They're unpaired. There are 3 of them, and they are right in front. And their names are, from superior to inferior, the celiac, the superior mesenteric, and the inferior mesenteric, which always confuses me. How can this be superior when it's not the most superior collateral ganglion? Why did they name it this? Just because they hate us and they want to confuse us? Anatomists aren't beyond that, but did they have a better reason for these names?
Because probably the 2 mesenteric have similar function, possibly.
Close. You got the right idea. It's not function, but actually structure. It turns out that these ganglia are named for the blood vessels they sit next to. As we know, blood vessels and nerves tend to go in the same locations, and these ganglia are located right next to blood vessels. The celiac is right next to the celiac trunk, which goes to the liver, the stomach, the pancreas, the spleen, organs like that. And then we have 2 blood vessels that go to the intestines, the superior mesenteric and the inferior mesenteric arteries. And so not surprisingly, the 2 ganglia right next to them are the superior mesenteric and inferior mesenteric ganglia. So absolutely, you have the right idea. It is because of the blood vessels they sit next to. They're not named based on their conditions relative to each other, They're named by the blood vessels they're sitting next to. So the celiac is the most superior, then superior mesenteric, then inferior mesenteric. And notice also, as you can see from this illustration, they innervate different organs of the body. And we'll go ahead and do this here. We'll come back and do this again in a minute, but our celiac, as you can see from the illustration, innervates the liver, the gallbladder, the stomach, the spleen, the superior mesenteric, innervates all of the small intestine and the proximal part of the large intestine, and our inferior mesenteric innervates the urinary system, the reproductive organs, and the distal large intestine, primarily the rectum and the anus. So notice, again, we can follow these pathways. Notice the nerves coming into the inferior, go to the reproductive organs, go to the urinary organs, and go to the rectum and anus. Those from the superior mesenteric go to the small intestine and the proximal part of the large intestine. And then those from the celiac ganglion go to the liver, the gallbladder, the stomach, the pancreas, and the spleen and pancreas. Obviously, as you can see, there is some overlap of this because, as we know, this is broad, elaborately branching. But in general, these are the three main pathways of the places that they go to. So notice, while for the most part, this is a teeny bit of a mess, if we go back kind of like we did with the parasympathetic in the last class, again, when it comes to where the pre-ganglionic neurons are, it's pretty straightforward. All of our pre-ganglionic cell bodies are all located from T1 to about L2 or 3. So there's no trick to that there. They're all located there. They are going to synapse. So then again, we'll worry about what the preganglionic axons do later. We'll do that in a different way. But what I do want to talk about is then what our postganglionic cell bodies are located. And they're both basically located in one of two locations. They are either in chain ganglia or they're in collateral ganglia. With the collateral ganglia, there are three and only three: celiac, superior mesenteric, and inferior mesenteric. For our chain ganglia, there's really three kinds we worry about. We have our superior cervical, we have our middle and inferior cervical, and we have the rest of the chain, which is basically T1 through the coccygeal. And this is important because we know, again, we'll worry about the pathway later. We'll worry about those later. We'll just leave that blank for now. But what we can talk about is what effectors they innervate. So our superior cervical chain ganglia, what effectors are innervated by the superior cervical chain ganglia? Come on, we just went over this. Yeah, excellent. I love that. That's a great way of describing it. All smooth muscle and glands of the head. Perfect. I'm gonna run out of room here, so let's actually we can make this work. Perfect. You know, just don't make it a little smaller. There we go. Excellent. What does the middle and inferior cervical chain ganglia innervate?
Isn't that like the heart and the lungs?
Exactly. Absolutely. And what does the rest of the chain ganglia innervate?
Some of the proximal, or the small intestine and the proximal large intestine.
Nope, that's down in the collateral ganglia. That's below the diaphragm.
Was it the skin?
Yeah, and I'm actually going to steal from Mitch because I like the way she wrote that. All the glands and smooth muscle of the skin. Perfect. Now, remember our collateral ganglia are the ones that go below the diaphragm. So someone mentioned the small intestine and the proximal large intestine. Which of our three collateral ganglias were the ones that innervated all of the small intestine and the proximal large?
The superior mesenteric.
Excellent. So then what did the celiac ganglion innervate?
The liver, gallbladder, spleen, pancreas.
And one more.
The stomach.
Stomach. There we go. Excellent. Perfect. Excellent. And then what does that leave for the inferior mesenteric?
reproductive system, the urinary system,
and yeah, distal large intestine. Or if you don't want to say distal large intestine, you can just say rectum and anus. Right. Basically, the structure is involved with defecation. Once again, defecation, reproduction, and urinary system. Excellent. So essentially, at its core, this is what we're looking at. Chain ganglia and the collateral ganglia innervating different organs. Now, obviously, the pathway's got to be a little bit more, you know, because we could just, in simplicity terms, know that all of this just goes like that. But of course, it's not going to be quite that straightforward. So we are going to need to go back and figure out what structures are made by the preganglionic axons, what structures are made by the postganglionic axons, and be able to draw those out. But at its core, this is what we're looking at. These are the sympathetic ganglia, and these are the effectors that they innervate. All right, so as a simple starting point, this is a good reference to remember as we're working our way towards those pathways. All right, there we go, and we just did that already. Excellent. Now, the good news is, as I mentioned, there are going to, using this data that we just put on that last table, there are going to be 4 sympathetic pathways that we will describe. And like I said, I guarantee one of these is going to be an essay question on the exam. And in fact, if we were in the classroom, I would make you draw them. Since we're not in the classroom and that whiteboard isn't the best tool, I'm not going to force you to draw these, but you will need to describe them in words. However, I will still illustrate them because I think seeing them is a good way of doing this. Now, I want to make a point of emphasizing that your book does indeed describe all 4 pathways, but it really only emphasizes 3 of them. And I don't want you to forget about that 4th. So what we are going to do is we are basically going to draw this, identify the structures, and then once we do that, we will write out our pathways to those 4 possible pathways that could be potential essay questions. So for that, let's go to our whiteboard. Perfect. Starts easy. We know we have to start here. Put this at the top. All of our sympathetic pathways start in the spinal cord. Now, seeing that the sympathetic pathways are all motor, do we really technically have to draw the dorsal root and the dorsal root ganglion to describe these pathways? No, because that's sensory. We know only sensory travels up it, but right, for consistency, we'll put it there just for the heck of it. There's our spinal nerve where it comes together. And then as we know, it's going to branch not once, not twice, but 4 times. Excellent. So let's stop right here for a second and do some quick labeling. This, of course, is our ventral root. This, of course, is our spinal nerve. Let's cheat and put that up there. This, of course, is our dorsal ramus. This, of course, is our ventral ramus. This is our white ramus. Oh, you know what? I'm going to cheat. Hold on a second. There. White ramus. Gray ramus. Literally no new information here. All right, this is stuff that we already know, we've already identified, we've already talked about. And we now know that this white and this gray ramus connect to our chain ganglion. And I think my chain ganglion's a little big here, so let's cheat and make it a little smaller. Now, speaking of cheating, again, you know I'm not a good artist, but we know that this chain needs to be connected to others. And again, if I was a good artist, I'd be able to draw this as a nice three-dimensional structure. I'm, of course, not able to do that, so I'll cheat and draw them off to the side. We know that it's going to connect to a chain ganglia that is above it, and we know that it is going to connect to a chain ganglion that is below it. So this would be our ascending chain, and this would be our descending chain. And again, you're just going to have to live with my horrible artistic skills to make some sense of that. However, we also know chain ganglia are not the only ganglia. We also know that here in front of the spinal cord are going to be the unpaired collateral ganglia. Or since it's just one, we'll make it a ganglion. There we go. We have our collateral ganglion located here. In front of our spinal cord. And again, there's no new information here that we've drawn. All we've done is illustrated in this simplistic form the structures we're responsible for. And notice if we cheat and go back to that illustration, notice they've shown us the exact same thing. Notice here on this illustration, we have our spinal cord. Let's not use green since they use green. We have the dorsal root and the ventral root to the spinal nerve. We have our dorsal ramus, we have our ventral ramus, we have our white ramus, we have our gray ramus, we have a chain ganglia, we have the ascending and descending chains, and here it actually shows them connecting to other chain ganglia. And it shows us a collateral ganglia in the front. Now notice it also shows us that there's a structure connecting those two, and we haven't identified that yet. But even though we haven't given it a label, let's go ahead and draw it on here because we know we're going to need it eventually. Speaking of cheating, I'm going to put my chain ganglion up there. So we know there's got to be some connection between those. So we'll go ahead and draw that. And we'll worry about labeling what it is in just a minute. So this right here is going to be our starting point. All right. Questions on that? All right. So what we are going to do is we are going to identify the potential effectors we want to innervate. And as I mentioned, there are going to be 4 possible pathways. And that I guarantee you will be, one of these will be an essay question on the exam. All right, we have set the stage, but I want to come at this fresh, and we've been going for an hour. So I think this, well, you guys had your quiz, but still, this is a good natural stopping point. So let's come at this information fresh. So let's go ahead and take our first break here. We will go ahead and take a 15-minute break. We will restart at 9:15 or 9:17, and we'll start the recording at that time. All right, any questions? All right, I will see you guys in 15 minutes. Any questions before we get started? All right, so let's talk about our 4 pathways, and we'll start with pathway 1. Pathway 1 is going to be to any of the structures of the skin. So, and again, I like this can be, make this a little smaller. So again, this can be sweat glands, this can be the erector pili muscle. Etc. Basically any structure that I were to give you associated with the skin. Now, one of the easiest ways to think of these pathways is to start with what we know. Where are the preganglionic neurons located? Let's steal a little from down here. We'll do it in blue. What do we know about the preganglionic neurons in the sympathetic nervous system, just period? In general, what do we know about them?
They're myelinated.
Excellent. Their axons are myelinated. What else do we know about them? Multipolar. Excellent. I can think of 2 more good things that we know about them. Where are their cell bodies always located? Excellent. And what do we know about the neurotransmitter that they release? What neurotransmitter do all preganglionic neurons release? Acetylcholine, excellent. And is it excitatory or inhibitory?
Excitatory.
Excellent. Perfect. So let's slide this over here for right now. And we can do that. We can draw our multipolar neuron that starts in the lateral gray horn. Its axon needs to leave the spinal cord. So does it leave out the dorsal root or the ventral root?
The ventral.
Ventral root, excellent. How did you know?
Because dorsal is where the sensory comes in.
Right, absolutely. And notice we didn't label the dorsal. We only labeled the ventral. That makes it easy. But no, you're absolutely right. We know that the ventral root is where our motor axons go to the spinal nerve. And as we mentioned, this is going to be myelinated. Perfect. So we know the beginning of our pathway. Starts with our preganglionic neuron, whose cell body is located in the lateral gray horn. Its myelinated axon leaves the ventral root, goes to the spinal nerve. Now, for this effector, we know that it takes two neurons to get to any of the effectors in our sympathetic pathways. So which ganglia are we going to synapse in? Chain or collateral? Which ones innervate the skin? Collateral ganglia or chain ganglia?
Chain.
Yeah. So we know our postganglionic neuron, and let's write what we know about our postganglionic neuron. What do we know about it? Let's use the same format. Axons are what? Are axons of our postganglionic neurons myelinated or unmyelinated? Unmyelinated, excellent. Is it multipolar like our preganglionic? Uni? It's still a motor neuron, so it is still going to be multipolar. So it is going to be a multipolar neuron. The cell bodies are going to differ in their location, so we won't worry about that right now. But let's talk about neurotransmitter. What neurotransmitter do most release?
Acetylcholine.
For the sympathetic, do most release acetylcholine?
No.
So what was it that they released? Norepinephrine, excellent. Excellent. And again, this will be either excitatory or inhibitory based on the effector. All right. So those are the things we know about the preganglionic and postganglionic neurons. And we now know that for our first pathway, we are going to need a multipolar neuron whose cell body is located in one of these chain ganglia. All right. With me so far? Excellent. Stunned silences, of course, mean yes, go faster. So let's do that. Excellent. So we need our preganglionic neuron to synapse on our postganglionic neuron. And notice this myelinated axon has two paths that it could take. it could either take the gray ramus or the white ramus. And being a myelinated axon, which of these two do you think this axon will help to form? Do you think a myelinated axon is going to help to form a gray ramus or help to form a white ramus? Well, let's see. Myelinated axons tend to be what form the white matter in the brain and spinal cord because that myelin is all that fat and it has a whitish color. So clearly a myelinated axon is going to help to form the gray ramus, right? No, Sarah's got it. Absolutely. It is going to travel down the white ramus to synapse on our postganglionic neuron. Now, remember, these things branch elaborately. So it's probably going to ascend, it's probably going to descend, it's going to go to all these different places, but it is going to synapse on a chain ganglia. And of course, as we mentioned, it is going to release acetylcholine, and that acetylcholine is going to be excitatory. Questions on that? All right. We formed our synapse, but now our goal is to get to the skin. And if you remember before, we talked about how the dorsal and the ventral ramus, the dorsal ramus goes to the skin of the back and muscles. Of the back. The ventral ramus goes to the skin and the muscles of the trunk and the limbs and the anterior side of the body. So if we need to innervate some structure of the skin, we probably need to get the axon of this postganglionic neuron, which just happens to be unmyelinated. We need to get it back to the dorsal and the ventral ramus. And might a path where an unmyelinated axon could travel exist? And what would that be? The gray ramus, absolutely. So this axon is going to travel out the gray ramus to either the dorsal ramus or the ventral ramus, where it will get to some effector of our skin. And of course, once there, what neurotransmitter is it going to release? Norepinephrine, excellent. And again, depending on what it is innervating, it could be excitatory or inhibitory. Right? If it was going to a sweat gland, it might be excitatory, stimulating that sweat gland to produce sweat. And there, just that easily is our first pathway. Notice if we wanted to do this in words, we could come back here. Right? Essentially, what we have described right here is this pathway. And let's go with purple for this first one. Any of the glands and smooth muscles of the skin. Again, as we know, we start with a pre-ganglionic neuron whose cell body is located in the lateral gray horn of the spinal cord. And what are the structures now that we can say that its axon forms? When it leaves the spinal cord, what is the first thing that it becomes a part of? Ventral root. From there, where does it go? Spinal nerve. Then white ramus, where it then synapses on that postganglionic neuron, releases acetylcholine, excites the postganglionic neuron. And that postganglionic neuron, where does its axon go?
Gray ramus.
To? Where does the gray ramus take it?
The dorsal ramus or the ventral ramus.
Which then, of course, takes us to the skin, where it most likely releases norepinephrine, and then again is either going to be excitatory or inhibitory in its effect. And so now we've done it with the pretty pictures, and now we've done it with words. Questions on this? Again, if this doesn't make sense, it's only going to get worse from here. So now is the time to say, does this make sense?
Would you want us to explain how the pre-ganglionic neurons and the post, like their attributes, so like the axons are myelinated, they're multipolar, like all of that within the explanation as well?
Yes, absolutely. And notice that's going to be true for all the pathways. So that's why I didn't bother emphasizing it as part of the pathway, because the good news is the preganglionic neuron is always going to be in the lateral gray horn. Its axon is always going to be myelinated. The postganglionic is always going to be unmyelinated. The pre always releases acetylcholine. The post is, for our purposes, always going to release norepinephrine. So yeah, so those aren't going to change no matter which pathway we're talking about. Okay, so great point.
Yeah.
All right, excellent. If we are good with pathway 1, then let's do some erasing. Oops. And do pathway 2. Oops. Before I do any more, let's use orange for pathway 2. Pathway 2 is going to be internal organs above the diaphragm. This, of course, means the heart and lungs, but this also could mean salivary glands or the smooth muscle in the eye, etc. Notice I specifically separated these two because when we're talking about going to an internal organ above the diaphragm, what type of ganglion are we going to synapse in, a chain or a collateral? Which ones goes to organs above the diaphragm, the chain ganglion or the collateral ganglion? Remember, collateral go below the diaphragm. So if we're going above the diaphragm, it is the chain ganglion. And which chain ganglion specifically goes to the heart and lungs, or chain ganglions, plural, go to the heart and lungs? Middle and inferior. Cervical. And which ganglion specifically goes to those internal organs in the head? Excellent. So we can be precise in ours. Now, notice our goal again in pathway 2 is going to be to get to and synapse in the chain ganglion. So do I actually have to change my pathway for the preganglionic neuron at all? Didn't we synapse in a chain ganglia for pathway 1?
Yes.
We're synapsing in a chain ganglion pathway 2. So notice for pathways 1 and 2, our preganglionic neurons pathway is identical. We don't have to change anything. So that's convenient. Let's cheat and go back here. Pathway 2, we'll do an orange. So again, pathway 2, we are either going to the heart and the lungs, or we're going to the smooth muscle of the head. We're of course starting in the lateral gray horn of the spinal cord. And notice the pathway is exactly the same for this. Ventral root to the spinal nerve, to the white ramus. Completely identical to the previous one. The only difference is now we can be specific. If we're going to something inside the head, we would want to say cervical. If we were going to the heart and lungs, we'd want to say middle and inferior cervical. So instead of just saying generic chain ganglion, we would want to specify specifically which chain ganglion we are going to. All right. But other than that, notice it's the exact same pathway. And in fact, we are still going to release acetylcholine. And it is still excitatory. However, here our postganglionic neuron needs to get to the heart or the lungs or the salivary gland. To do that, it is actually going to leave out of a different pathway from the chain ganglia, one I hinted at with the hole that I put on the chain ganglia, but one I hadn't identified yet. And these axons are going to leave the chain ganglia to either go to the heart and the lungs, or to go to some organ of the head. And the structure they form when they leave is what is known as the sympathetic nerve. And of course, when it gets to its destination, what neurotransmitter does it release?
Norepinephrine.
Norepinephrine. And again, it's going to be excitatory or inhibitory, depending on what the effector is. All right, if it's your lacrimal gland, then it's going to be inhibitory, drying out your eyes. If it's your heart, it's going to be excitatory, making it beat faster. And so notice we can now go back to our previous picture and we can fill the rest of it in with the words. From the superior cervical or the middle inferior cervical, we form sympathetic nerves. And those sympathetic nerve goes to either the head or the heart and lungs and releases norepinephrine, which can be excitatory or inhibitory. And that is pathway 2. Questions on that?
So for the superior, middle, and inferior cervical chain ganglia, they technically have the same pathway. The only difference is where their effectors go to.
Exactly.
Okay.
Yeah.
So the only difference is like one can go to the heart and lungs and the other goes to the smooth muscle and the glands of the head.
Exactly.
Okay.
Which is why they're clumped together into the same pathway. Because notice the only difference is which effector and which ganglion, but it's still a chain ganglion and it's still a specific effector. And in fact, we see that with pathway 3. Pathway 3 is basically to all of the organs below the diaphragm. Now, notice when we're going below the diaphragm, there's lots of different locations we can go to, and you're going to have a choice of 3 collateral ganglia. So the pathway is the same. It's going to be one pathway we take to a collateral ganglia, one pathway we take from the collateral ganglia to the organ. The only difference is, depending on which organ it is, can you tell me specifically which ganglion it is? So if I tell you to give me the pathway to the stomach, which collateral ganglion are you going through?
Celiac.
If I'm telling you you're going to the bladder, which collateral ganglion are you going through?
Inferior mesenteric.
Yeah, and if I say you're going to the small intestine, which collateral ganglion are you going to?
Superior mesenteric.
Yeah, superior mesenteric, exactly. So the pathways are identical. The only difference is which collateral ganglia you're using. Okay, excellent. So perfect. We now know what our third pathway is going to be. Pathway 3 is organs below the diaphragm. Oh, I don't want that capitalized.
If there's only 3, how are there 4 possible pathways?
Ah, that is a mystery, isn't it? Well, let's figure out 3 before we worry about 4. But I like how you're thinking. That's spectacular. Excellent. So let's make it easy. We know that these are all going to collateral ganglia. So let's be specific. Celiac, if it's the celiac, what would the organs be? Liver. What else?
Gallbladder.
Gallbladder. I heard stomach.
Spleen.
Spleen. And
Pancreas.
pancreas. Is that 6? 2, 3, 4, 5. No, we're missing one. What are we missing? liver, gallbladder, stomach, spleen, pancreas.
The kidney?
No, not kidney. Are there 6? Or are there only 5?
I don't think there's.
Liver, gallbladder, stomach, spleen, pancreas. No, I guess you're right. I guess there's just 5. My bad. 5. All right, excellent. Superior mesenteric. What would that be the collateral ganglion for?
The small intestine and the proximal portion of the large intestine.
Excellent. And then the inferior mesenteric. Repro, urinary, and rectum and anus. Excellent. All right, perfect. We know where we need to go. Let's figure out how we're getting there. Now, the good news is, once again, there's really only one way to start with the preganglionic neuron. Oops. We are on pathway 3 now. There's only one way to get where we need to go. It can only come out the ventral root. The ventral only can go to the spinal nerve. And being a myelinated axon, it can only go down the white ramus. However, this time, do we want to synapse in the chain ganglion?
No.
We want to synapse in a collateral ganglion. So what's going to happen is this myelinated axon is going to keep on going, leaving the chain ganglion without synapsing and going to the collateral ganglion. So this myelinated axon enters the chain ganglia, but then leaves the chain ganglia. And when it leaves the chain ganglia, it forms a structure known as the Splancnic Nerve. If you remember, in the parasympathetic, we had pelvic splanchnic nerves. So P for pelvic splanchnic for parasympathetic, S for splanchnic for sympathetic. So this is a sympathetic nerve, a splanchnic nerve. It is a sympathetic nerve, a preganglionic myelinated axon that leaves the chain ganglion, going to the collateral ganglion, where it will synapse. And of course, as we know, when it synapses, it is going to release acetylcholine, and it is going to be excitatory. It, of course, needs to synapse on our postganglionic neuron, multipolar neuron, unmyelinated axon, and this unmyelinated axon, I guess I have to cheat and sneak my way down here to get to our destinations, is an unmyelinated axon. And when it leaves, the axons that leave, once again, are called sympathetic nerves. Notice whether you're above the diaphragm or below the diaphragm. If you're inside the body and you're a postganglionic neuron, you form a sympathetic nerve. And of course, once it gets to its destination, it is going to release norepinephrine, and again, will be excitatory or inhibitory, depending on the organ. We've done it with the drawings. Let's do it with the words. Notice it starts the exact same way. Pre-ganglionic neuron, cell body located in the lateral gray horn. Its myelinated axon forms a ventral root, forms spinal nerve, and forms the white ramus. It starts the exact same way. However, this time it does not synapse in the chain, and instead it continues and forms the Splancnic nerve. And it is the Splancnic nerve that goes to one of our 3 collateral ganglia, releases acetylcholine, and excites our postganglionic neuron. The axons from our postganglionic neurons then leave, and they form a sympathetic nerve. And go to one of our organs below the diaphragm, where it releases norepinephrine and can be excitatory or inhibitory. And that is our 3rd pathway. Now, I've done a truly amazing job of drawing these things because I'm just that kind of an artist. And now we have it in the pretty words, but I also really enjoy beating dead horses. So let's go over this again with all of the pretty pictures and all of the words from your textbook as well to make sure we understand these 3 big pathways. And again, there are lots of ways to think about this to help you to organize the information. One way we can think about it is, where does the synapse occur? Where is the synapse between the preganglionic and postganglionic neuron? Well, notice the first place where that synapse could have taken place was in the chain ganglion. So remember, we start with a preganglionic neuron who's multipolar. Cell body is located in the lateral gray horn. Its myelinated axon leaves the ventral root to the spinal nerve. And then from the spinal nerve forms that white ramus, which again, remember, is the lateral one. It is then going to synapse in that chain ganglia. And remember, it branches elaborately. So it can ascend first, it can descend first. It probably does all 3. But the point is, it is going to synapse within that chain ganglia. Then, if we're going to the skin, our unmyelinated postganglionic axon, and again, this is another multipolar neuron. Its unmyelinated axon goes out the gray ramus to the dorsal and ventral ramus. And from there, as we mentioned, it goes to all of the smooth muscle and glands of the skin. Very simply, very straightforward. This is our first pathway. Questions on this? Now notice pathway 1 is almost identical to pathway 2. We have a multipolar preganglionic neuron. The cell body is located in the lateral gray horn. Its myelinated axon leaves the ventral root, forms the spinal nerve, and then forms that white ramus, that lateral communicantus, where it enters the chain. It can travel up, it can travel down, but it is going to synapse within that chain ganglia. And it synapses on that postganglionic neuron. That multipolar postganglionic neuron's unmyelinated axon now leaves, only here is the big difference. This time when it leaves, it forms a sympathetic nerve. It goes out a different pathway, out a different structure, because it's going to a different location. What is the locations, plural, that it is going to go to if it comes out the sympathetic nerve? You can either give me the general term for them, or you can give me specific examples.
The heart and lungs.
Excellent. Heart, lungs, eye, lacrimal gland, etc. Or, as who was it that said it? As Mitch pointed out, internal organs above the diaphragm. This right here, what the axon of our postganglionic neuron forms and where it goes, is the only difference between pathway 1 and pathway 2. And we've done it in words. You're going to be responsible for doing it in words, but your book does have some nice pictures. This one's actually not from your textbook, but I like it because it shows both pathways side by side. Again, remember, one side doesn't do one thing and the other side does the other. They've just shown it to us this way so that we can see them more clearly. Notice for both pathway 1 and pathway 2, what the preganglionic neuron does is identical. Starts in the lateral gray horn, ventral root, spinal nerve, white ramus, synapse. Lateral horn, ventral root, spinal nerve, white ramus, synapse. The difference is what happens to the postganglionic axon. To get to the skin, it synapses, leaves the gray ramus, and goes to the dorsal or ventral ramus to go to the spinal nerves, to go to the skin. And if it's going to go to an internal organ above the diaphragm, after the synapse, its unmyelinated axon leaves the sympathetic nerve to go to an organ above the diaphragm. Notice, as I mentioned, this picture is not from your textbook, but your textbook shows these pictures as well. Notice here it shows the pathway to the skin. Pre-ganglionic neuron. No, it's not use green. Pre-ganglionic neuron, cell body located in the lateral gray horn, ventral root, spinal nerve, white ramus, synapse. Postganglionic neuron, gray ramus to, in this case, the ventral root, going to the skin. And again, remember, your book also emphasizes the fact that it can ascend before it synapses or descend before it synapses as well. But it's still synapsing in the chain ganglion. It's still going out the gray ramus. It's still going to the skin. Any questions on our first two pathways? All righty. Now, notice with our first two pathways, our synapse between our pre and our postganglionic neuron takes place in a chain ganglion, but it doesn't have to. the synapse can also take place in the collateral ganglion. And notice the path starts the same way. We start with that preganglionic neuron that's located in the lateral gray horn, comes the ventral root to the spinal nerve, to the white ramus, to the chain ganglion. But notice this time it doesn't synapse in the chain ganglion. It continues past the chain to form that splanchnic nerve. And that splanchnic nerve is what travels to our collateral ganglia and synapses there. Still releasing acetylcholine, still excitatory, but now it's in a collateral ganglion. From that collateral ganglion, our unmyelinated postganglionic axon leaves, and again, forms a sympathetic nerve. And these innervate the organs of the abdominal pelvic cavity, the organs below the diaphragm. Notice here we see this picture again, not from your textbook, but I really, really like this because again, it reminds us that our collateral ganglia are unpaired, but it clearly shows this pathway. Pre-ganglionic neuron, cell body located in the lateral gray horn, ventral root, spinal nerve, white ramus, through the chain ganglion, out the splanchnic nerve to the collateral ganglia where it synapses. Then our postganglionic neurons unmyelinated, and it leaves to form that sympathetic nerve innervating some organ below the diaphragm. Questions on our third, let's say on the exam. No, great question. That's an excellent question. So here is exactly how I'm going to ask this question on the exam. Well, okay, not exactly. The wording is going to be very similar to this. Shouldn't have been all in caps, but you get the idea. Describe, you're going to see something where it says, describe the sympathetic pathway for the innervation of X. And X could be anything. Smooth muscle of the eye. It could be liver. It could be bladder. It could be sweat gland. Although I won't say sweat gland, I'll probably say sudoriferous gland. Etc. I will give you some specific organ. So I will give you the specific organ and you have to give me the specific pathway. And then of course the question will say, make sure you describe all the neurons, describe all the structures, describe all of the neurotransmitters, all of that kind of stuff as well. So if I gave you the bladder, then yes, you would want to say that it was specifically the inferior mesenteric ganglion. If it was the smooth muscle of the eye, you would want to say that it was specifically the superior cervical chain ganglion. So yes, so it won't be the other way around. Although I guess on the lab exam, I could point to the celiac ganglion and I could ask you to identify two specific organs that are innervated by it. So I could ask it that way on the lab exam. But on the lecture exam, I'm going to give you an organ and you're going to give me the pathway to it. Does that make sense?
And there's going to be one of these or one of the parasympathetic, or is it just one out of all of them?
But I am guaranteeing you that there are 4 sympathetic pathways, and I'm guaranteeing you that one of these will be a question on the exam.
Right.
Now, could there also be parasympathetic pathways? Sure, but those are part of a bundle of another bunch of questions. I'm guaranteeing you one of these 4 pathways are going to be here. So one of these 4 pathways, or one of these 4 pathways are going to be on the exam. Now, of course, what's the problem with that?
There's not 4.
We haven't done 4 yet. So any questions on these 3 before we finish? Oh, no.
No.
All right. Then let's do 4. What's left? Anyone have any idea what we haven't talked about yet? I can't think of any place we haven't gone. So what in the world is pathway 4? Well, if
The
you
adrenal gland?
exactly. If you remember, one of the keys to our sympathetic nervous system, one of the ways we get our big global effect is the fact that our sympathetic nervous system innervates the adrenal gland and tells the adrenal gland to release that adrenaline into our blood. Now, actually, I started this, but I'm going to go ahead and erase that for a second because we need to do some really quick math. Here is our kidney. And sitting on top of the kidney is our adrenal gland. Obviously, that's not drawn to scale. But what we do need to know is that our adrenal gland has a chewy nougat center and a candy-coated shell. When we have that type of an organization, and in a superficial area and a deep area, what do we typically call the superficial area?
Cortex?
There we go, cortex. And the inner region then would be what?
Medulla?
Yeah.
Medullary
Medulla,
cavity?
absolutely. It would be the medulla. It turns out that's exactly how the adrenal gland is built. And in fact, it is the medulla of the adrenal gland that actually releases the adrenaline. It's not the whole adrenal gland. It is actually the, oops. Yeah, we can leave it big to emphasize it. It is actually the medulla of the adrenal gland that actually releases the adrenaline into our body, giving us that big global effect.
Are we allowed to use that name? Because in the book they use a different name.
Medulla of the adrenal gland is acceptable. Adrenal gland is fine. The other thing, because this sits on top of the kidney, it is sometimes referred to as the suprarenal gland. And again, both of those are acceptable names for the organ. So whether you want to call it the suprarenal gland or whether you want to call it the adrenal gland, they mean the exact same thing.
All right, thank you.
Yep. And so that's our goal, to get to the chewy nougat center of the adrenal gland. Now, the good news is, once again, this pathway begins the exact same way all of our other pathways have begun. We start with a preganglionic neuron whose cell body is located in the lateral gray horn. Its myelinated axon leaves the ventral root, leaves the spinal nerve, enters the white ramus, enters the chain ganglia, and once again, does not synapse. So it helps to form the Splancnic nerve, where it leaves and goes specifically to the celiac ganglion. However, when it gets to the celiac ganglion, once again, it does not synapse. This myelinated axon actually goes all the way to the medulla. So it leaves the chain ganglia, forming the Splanchnic Nerve, goes to the celiac ganglion, leaves, forming a sympathetic nerve, and actually travels all the way to the medulla of the adrenal gland. Now, we still need a second neuron, and lo and behold, here in our medulla is our postganglionic neuron. It's located here in the medulla, and our postganglionic neuron then releases primarily epinephrine, about 80%, or again, you could use adrenaline into the blood. So it is in this fashion with this special fourth pathway that our sympathetic nervous system releases that adrenaline into our blood, giving us our big, huge global effect.
Does it also release norepinephrine?
Yeah, so notice
Okay.
80% epinephrine, so the other 20% is norepinephrine that it is releasing into the blood. But remember, for our purposes, they function the exact same way. So here, now we can add our final fourth pathway. Let's do this one in light blue. So again, we start in the lateral gray horn with our preganglionic neuron. It's myelinated axon, leaves, again, ventral root to our spinal nerve, to our chain ganglion, no synapse, Splancnic nerve, to our celiac ganglion, again, no synapse, space here, to the medulla of the adrenal gland. Where it then synapses on our postganglionic neuron, which notice isn't in a ganglion. Essentially, the medulla of the adrenal gland is the ganglion where our postganglionic cell body is located. And of course, it, as we mentioned, releases epinephrine and norepinephrine into the blood. And so if you think about it this way, if it's releasing norepinephrine and epinephrine into the blood, what are the effectors of this epinephrine and norepinephrine that are being released into the blood?
The entire body.
Yeah. Once it's in the blood, it pretty much goes everywhere.
So there really isn't a collateral ganglia for this last pathway.
Nope. It,
So
although
how-
it passes through the celiac collateral ganglia. So there is a collateral ganglia that way, but it doesn't synapse in one, no. And again,
Okay.
notice, like I said, Oh, here's your book's pathway for pathway 3. Again, suprarenal or adrenal, either of those are acceptable terms. I don't care which one you use. But it is the medulla of the suprarenal gland or the adrenal gland. Lateral great horn, ventral root, white ramus, don't synapse in the chain. Go to the celiac ganglion, don't synapse. Continue to the medulla, synapse there. Release our hormones into the blood and affect everything. And as I mentioned, your book doesn't have a pretty picture of it, but notice this one does, which is why I use these pictures. Notice our preganglionic neurons doing all the work here. Ventral root, white ramus, ventral root, spinal nerve, white ramus, through the chain ganglion, Splancnic nerve, through the celiac ganglion, sympathetic nerve, to the medulla, where it then synapses and releases epinephrine into the blood, and it goes everywhere. And that is our 4th pathway. And notice, if we come back to this pretty picture, this scary spider octopus that we saw, lo and behold, this shows us all 4 pathways. Pathway 1, lateral gray horn, ventral root, spinal nerve, white ramus, synapse in a chain ganglion. Black neuron is the postganglionic neuron, leaves the dorsal ventral root and goes to the skin. There is pathway 1. Pathway 2. Start in the spinal cord. Go ventral root, spinal nerve, white ramus to a chain ganglion, where we can synapse right away or we can ascend. Synapse in the chain, and our postganglionic neuron leaves out the sympathetic nerves to go to the internal organs of the head, go to the heart, go to the lungs. And that is pathway 2. Pathway 3. Start in the lateral gray horn, ventral root, spinal nerve, white ramus to the chain ganglion, and keep going to one of our collateral ganglia. Notice if it's the celiac ganglia, you're going to synapse on the celiac ganglion and go to the liver or the gallbladder or the stomach or the spleen or the pancreas. If instead you're synapsing in the superior mesenteric, you're going to the small intestine or the large intestine. And if you're synapsing in the inferior mesenteric, you're going to the reproductive organs, the urinary organs, or the rectum and the anus. And again, notice as you leave the chain ganglia without synapsing, you form splanchnic nerves. And that's pathway 3. And lo and behold, if you look really, really carefully, you can see how you could start in the preganglionic neuron with this red myelinated preganglionic axon, ventral root, spinal nerve, white ramus, chain ganglia, and through. Splanchnic nerve to the celiac and through without synapsing. Notice this red preganglionic axon goes all the way to the medulla of the adrenal gland, where it synapses in the adrenal gland and releases that adrenaline into the blood. So notice how this thing was big and scary when we looked at it the first time, but it actually does a really beautiful job of showing us our 4 possible sympathetic pathways. So as scary and intimidating as it may have been when you first looked at it, it's actually awesome. Questions on this? All right. I will leave you with this picture. And let's go ahead and let this information sink in. We're not quite done with this yet. We're going to beat this information up a little bit more. But let's go ahead and let this sink in. And then we'll come back at this and tackle this a couple more different ways just to make sure we understand it. All right. So any questions before we take our next break? All right. Let's take a 15-minute break. That means coming back at 10:36. And I will start the recording at that time. See you in 15 minutes. All righty. We have covered the 4 pathways, but like I said, I want to make sure to continue to emphasize this information because like I said, it is going to be one of your big essay questions. And I appreciate that it can be a bit confusing. So like I said, I think one of the important things to do is to try to think of in terms of all the different components, right? So like we talked about, when you get to that chain ganglia, right? When we think of what our sympathetic axon can do, as we mentioned, it always starts the same, out the ventral root to the spinal nerve, from there to the white ramus, and it enters. And then one choice it has is to synapse right away. One choice it has is to ascend or descend before it synapses. The third choice it has is to continue through the chain ganglia without synapsing, forming the Splancnic Nerve, where it will then go and synapse in our prevertebral or collateral ganglion. And remember, it can also continue to the celiac ganglion, travel through and go to the medulla of the adrenal gland. So again, notice one way we can think of this is what it is that our preganglionic neurons axon is doing. Another way to think of it, as we've talked about, are where the cell bodies are located. Remember, for our preganglionic neuron, that's super easy. All of the cell bodies are located in the lateral gray horn between segments T1 and L2 of the spinal cord. Where we see differences is where the postganglionic neuron cell bodies are located. Some are located in the chain ganglion. And if they're in the chain ganglion, then they're going to the skin or the organs above the diaphragm. If the postganglionic neuron is located in a collateral ganglion, then it's going to the organs below the diaphragm in the abdominal pelvic cavity. And as we mentioned, if our postganglionic neuron is located in the medulla of the adrenal gland, then it's releasing its hormone into the circulation and it goes everywhere. Again, no new information here. We're just trying to find ways to organize this information and make sense of it. Lastly, we can think about those sympathetic nerves, or really what we're talking about here are the postganglionic axons. If our postganglionic axons form the spinal nerves going out the gray ramus, then again, they're going to the skin, to the muscles via those dermatomes. If our postganglionic neurons form sympathetic nerves from the chain ganglia, then they're going to internal organs above the diaphragm. Remember, those Splancnic nerves, which are made out of prevertebral ganglia, go below the diaphragm and ultimately lead to the adrenal gland as well. And again, all of these are on your handouts, so I'm not too worried about taking the time to write this down. What I do want to emphasize, though, is this part right here. The last way, and I want to do this on the whiteboard. The last way I want to talk about organizing this information is notice one of the big highways or byways that is passing through this pathway is that chain ganglion. And if you think about it, there is, actually, let's do it this way. Move this down there, move this down there. Six pathways leading into or out of a chain ganglion. And so what I think could be useful to do, and also makes a really good essay question, is to identify the six pathways, either to or from the chain ganglion. And again, this doesn't need to all be in. So we want to identify the pathways. We want to say whether they're made up of pre or post ganglionic axons. And of course, if we know if it's pre or post ganglionic, then we also know if it's myelinated or unmyelinated. We want to know whether it's going into or out of the chain. And if it's going into, where is it coming from? And if it's going out of, where is it going to? So let's do that. Here we have a chain ganglion with its six pathways. Someone give me the name of one of the potential pathways, either into or out of a chain ganglia. Name one of the six structures that comes out of the chain ganglion.
Ascending or descending?
Excellent. Ooh, you started with the hard one first. So we have the descending chain, and that goes hand in hand with the ascending chain. All right. So are these ascending and descending chains? Because again, they're going to be the same if you think about it. With two chain ganglia, what is ascending for one will be descending for another. So actually the information for these are identical. So for that ascending chain, is it made up of pre or post ganglionic axons?
Pre.
Pre.
Wait, why pre though? I just guessed.
Well, you guessed right. You had a 50-50 chance. That's right.
Yeah.
Remember, when we here, let's go back to the picture. Oh, I've erased all of the pictures. But remember, when this preganglionic axon comes in, remember we talked about how it could ascend or it could descend before it synapses. So it is indeed a preganglionic axon. And of course, if it's preganglionic, is it myelinated or unmyelinated?
Myelinated.
And again, remember that's going to be true for the descending as well. What makes this one tricky is this going into or out of the chain ganglion?
Both?
Yeah, exactly. These are going to be both. Some of the axons will be ascending and some of them will be descending. So indeed, in fact, I've got to cheat and bring this down a little bit. These are going both into and out of. And of course, they're going into and out of to other chain ganglion. Excellent. That was actually the hard one. And now that the hard ones are out of the way, it should be easy from here. Spectacular. So give me another one. Give me another structure that comes into or out of the chain ganglion.
Oh, white Remy.
Excellent. Our white ramus. Let's answer the questions. Is it made of pre or post ganglionic axons?
Pre.
So then is it myelinated or unmyelinated?
Myelinated.
Coming into or out of the chain?
Into.
Excellent. It is coming into the chain. If it's coming into the chain, where did it come from?
The ventral root.
True. Or we can even go back even further than that. How about the lateral gray horn? Right? Started in the lateral gray horn, but you're right. It wouldn't be wrong to say the spinal nerve. It wouldn't be wrong to say the ventral root. All of those would be acceptable places that it is coming from. And let's go easy. If we have the white ramus, what else do we have?
The gray.
Pre-ganglionic or post-ganglionic axons?
I guess each time, so I don't know. Pre.
The post.
Post. Why is it post? Maybe I should ask the question, why is it gray?
Because it's not myelinated.
Yeah. And if they're unmyelinated axons, can they be pre-ganglionic?
Nope.
No. Coming into or out of the chain?
Out of.
And going where?
Effectors.
Yeah, which effectors?
The skin.
Bingo.
So this is basically just collectively kind of almost
Yeah.
a summary of everything we just did.
Exactly.
Okay.
White ramus brings axons in, is pre-ganglionic axons coming in. Gray ramus is post-ganglionic axons going out. Two-thirds of the way done. Give me another structure coming into or out of the chain ganglion.
Ventral maybe?
Well, if we have to cheat, you can always come back to the picture. Notice here's our chain ganglia. We have identified the gray ramus and the white ramus and the ascending chain and the descending chain. So there's two more structures left. Excellent. There we go. Sarah's given us a good one. The Splancnic nerve. Excellent. That Splanchnic nerve, is it made up of pre or post ganglionic axons?
Pre.
Mm-hmm.
Yeah, I keep guessing. I don't know. Like.
Well, but if you're guessing
Wow.
right, then clearly you do know.
Okay.
Remember, when it comes in the white ramus, if it doesn't synapse, then that preganglionic neuron keeps going.
Okay.
And of course, that preganglionic neuron is myelinated. And where's it going to?
The collateral ganglia.
Yeah.
There you go.
Let's see.
Excellent. Going to the collateral ganglion. And that gives us one more structure. What is that last structure coming out of the chain ganglion?
Sympathetic.
There you go. Sympathetic nerve. Pre or post ganglionic?
Post.
Excellent. So does that make it myelinated or unmyelinated?
Unmyelinated.
Coming into or out of?
Out of.
Going where?
Thoracic cavity. No.
Yeah,
Organs
absolutely.
of the-
Excellent. To those internal organs above the diaphragm, like the thoracic cavity, like the salivary glands, like the, lacrimal glands, like things like that.
So for why wouldn't I think there was like a ventral and dorsal, I want to say ramus, I don't know if that's correct.
Yeah.
Why are they not considered a pathway?
Well, they could be if you wanted to, because, well, first, they don't connect directly to the chain. So
Okay.
which of these 6 structures goes to the dorsal and ventral ramus?
The gray, white.
Yeah. You're right. We could have put dorsal and ventral ramus here because those are the immediate structures that the gray ramus goes to, or we could have said its ultimate destination, which was the skin.
Okay.
Yep.
Thank you.
Perfect.
I have a question that's kind of random.
Okay.
When we go back in person, is the testing that you like, so I know right now we're doing it on the computers, are you going back to paper versions of the test?
Yes. God,
Okay.
yes.
Okay.
Yeah.
But it's virtually kind of the same format?
Yeah, essentially,
Okay.
like I said, it's still going to be multiple choice questions and essay questions. It's still going to be in fact, for the lab exam, some lab exams will actually be a PowerPoint presentation that will be on the screen. Some will be station-based, where you're going station to station. Like, for instance, the bones or the muscles would have been station to station, but the tissues, epithelial tissue and histology tissues, those are the slides that basically would have been identical to the one. So it'd just be a PowerPoint presentation that we would all go through together on the board for that. So yeah, so again, I've for better or for worse, I've tried to keep the format of this as close to what it would be in the actual classroom, so that you're getting as close to the classroom experience as you would get.
Okay, thank you. And my other question is,
I'm
I know
sorry,
there's a
Jackie,
new.
your quota.
Oh, okay.
No, I'm just kidding. What's your question?
I know there's another variant of COVID. Do you think that's going to change anything in regards to going back in person? Like, it just came out, and I
Yeah,
think
I
even
know.
today there's
have.
like a spot of it in California. So I don't know.
Oh, really? I hadn't heard that as of yet.
It was like 5 minutes ago. I just showed up in my.
I don't know. I have long since given up trying to guess how things will change, because I'm never right. So as of right now, my expectation is that they will keep things the way they are. You are going to be required to be vaccinated or to have the weekly testing if you get the waiver, and we'll be in the classroom. Now, the one thing that might change that I could see potentially is, as of right now, because of the testing and because of the vaccination mandates, as of right now, they weren't planning on having any other requirements. So you weren't going to be required to social distance or mask or anything like that in the classroom, because of the expectation that everybody is safe. I don't know if Omicron turns out to be something more serious, if they may change that, if they may go to masking, or if it becomes really bad, then it's possible that we could be, and God, I hope it doesn't happen, but anything's possible. Could we be back online? Possibly. I have no idea. I you know as much as I do. In fact, you know more than I do, because last I heard, the closest it had gotten to us was Canada. I hadn't heard that it had been, although I'm not shocked that Omicron's in the United States, but yeah, so I'm in the same boat that you are. We'll wait and see what happens. Nobody's told us anything.
Okay, thank you.
Yep. All righty. On that depressing note, again, I don't want to put anybody at risk, but clearly this is not a class that should be taught online. This is a class that we should be in the classroom. We should have been able to touch and taste the bones and do all of those kind of things. And so I'm looking forward to being back on campus, and I wouldn't say I'm hopeful or optimistic because my hope and optimism have been crushed months ago. But the expectation is to be on campus, and we'll see what happens. Until they tell me otherwise, that's what I'm expecting.
Thank you.
All righty. Excellent. So we did that. We did all of that. And that is our sympathetic and parasympathetic pathways. And again, these illustrations do a nice job of showing all of that stuff. All right. The good news is that is the end of the heavy lifting. We still have a little bit more information that we need to cover, but that is the end of the heavy lifting. There's just some additional information and really some vocabulary we're going to be adding. For instance, we know our sympathetic and parasympathetic nerves are going to similar locations. Both the sympathetic and parasympathetic are going to the heart or going to the stomach or going to the bladder. So not surprisingly, they form elaborate networks of nerves. And what's the fancy word we give for an elaborate network of nerves?
Plexus.
Plexus, absolutely. They form plexes. And again, just like the ganglia, they tend to lie along large major blood vessels, and that's pretty much where they get most of their names. So cardiac, celiac, inferior mesenteric, hypogastric, all of these are examples of plexes, and your book has this great picture that does a really nice job of showing this. Notice if we look at this illustration, we can see the fancy chain ganglia coming down and coming up the other side. So we can see that. We can see our 3 collateral ganglia located here on the midline and all of the elaborate mesh of nerves. And in fact, if we get the pesky person out of the way, we can see these things even more clearly. Now, I am not going to give you this illustration on the exam, and I'm not going to have a big arrow pointing, for instance, right here, and have you try to figure out that that is the inferior mesenteric plexus. However, while I won't give this to you on the exam, there is one plexus I did want to mention to you, and it is this one, the celiac plexus. The celiac plexus is located, right, notice right here, right below the diaphragm, where the celiac ganglion is located, where the superior mesenteric ganglion is located, and many sympathetic and parasympathetic nerves come into this structure, including one somatic motor nerve. This illustration doesn't show it, but your friend and mine, the phrenic nerve, also comes down and forms part of this celiac plexus. And what did the phrenic nerve do again? This is only 1 week from today. Well, okay. Maybe you've never heard of the celiac plexus before, but maybe you've learned it as one of its other 2 names. It is also known as your 4th chakra, or it is also known as your solar plexus. And if you happen to be standing on the field, not on the field, but on the sidelines at a soccer game, cheering for your friends, but really you're talking to your other friends. And while you're distracted and talking to your friends, you get hit right below the sternum by that soccer ball that has been kicked into your belly unexpectedly, putting pressure on that location, disrupting the activity of the celiac plexus, and thereby disrupting the activity of the phrenic nerve. What does the phrenic nerve do for you again?
Diaphragm.
It controls the diaphragm, absolutely. And when you disrupt that activity of the diaphragm, you can no longer contract your diaphragm. And suddenly breathing becomes a lot more challenging. Luckily, as we know, we have accessory muscles like the sternocleidomastoid, like the pectoralis minor, other ones that can help us to change the shape of the thoracic cavity and move air in and out. So we're not going to die, but we sure feel like we're going to die because it's a whole lot harder to get that air in and out. And of course, what do we call that?
Getting the wind knocked out of us.
Yeah. Getting the wind knocked out of you is caused by this disruption of the celiac plexus. Because the phrenic nerve forms part of the celiac plexus, when its activity gets disrupted, you get the wind knocked out of you. So again, most of you either had the wind knocked out of you or at least seen somebody else get the wind knocked out of you, and now you know why.
That can happen when you like land on your back too, right?
Yeah, absolutely. That pressure wave from the back can also disrupt. Again, basically, the pressure inside that abdominal pelvic cavity can disrupt the function of these plexes. And again, are you consciously aware of the fact that maybe your stomach is not functioning at its optimal or your kids' kidneys aren't functioning at their optimal? No, but when you can't contract your diaphragm optimally, you're definitely aware of that. So yeah, so it affects other things as well. It's just the effect on the diaphragm because of that phrenic nerve that is the most obvious. All righty. From here, we are going to focus not so much on new information, but new vocabulary. Basically, as we know, most neurons communicate to other neurons. So a neuron has an axon, releases a neurotransmitter, and that neurotransmitter binds to some receptor on the target cell. And like I said, most target cells are other neurons. Absolutely no new information there, right? And we know that one of the most common neurotransmitters that is released is acetylcholine. And so for that acetylcholine to have an effect on the target cell, it must have an acetylcholine receptor. And we know one other thing. Anatomists hate us. So instead of using the term acetylcholine, a neuron that releases acetylcholine, instead of calling it an acetylcholine neuron, we call it a cholinergic neuron. So a cholinergic neuron is just a fancy way of saying that it is a neuron that releases acetylcholine as its neurotransmitter. Absolutely no new information there. We're just giving it a new name. So any neuron that releases acetylcholine is a cholinergic neuron. And
So if we're using this, do you want us to use this on the exam, like when we're explaining, like releasing acetylcholine? Do you want us to say the cholinergic neurons release?
No,
okay.
But if I were to ask you to identify a cholinergic neuron, you need to know what that term cholinergic neuron means. And we've talked about 4 or 5. Yeah, we basically talked about 5 cholinergic neurons. So identify those in a minute. But before that, we need to add one more label. A receptor that responds to acetylcholine, we are going to call a cholinergic receptor. So again, there is literally no new information here. We have just added 2 new labels to what we already know. We know some neurons release acetylcholine. We know some receptors respond to acetylcholine. No new information there. We're just giving them fancy names because our anatomists love their fancy names. And as I mentioned, there are actually 5 different neurons that we've identified as being cholinergic. Notice all preganglionic, both sympathetic and parasympathetic neurons, both of those. So really 1 is 1 and 2. All autonomic preganglionic neurons we know release acetylcholine. We know all parasympathetic postganglionic neurons release acetylcholine. Now, most sympathetic postganglionic release what? Norepinephrine. But what's the key word in that sentence?
Most.
Most means not all. So remember, we did say, we did say that there were a few sympathetic neurons that are postganglionic neurons that will release acetylcholine. And it turns out it's not random. They're ones like to our sweat glands and to our blood vessels of our skin and muscles. Which brings us back to those magic mushrooms that we were talking, not the magic mushrooms, that's something totally different. Those poisonous mushrooms. Remember we were talking about those poisonous mushrooms that students of mine, boyfriend took. Remember they triggered a parasympathetic response by making their pupils dilate, making their airways constrict, making them make more mucus. And because they affect these acetylcholine receptors, remember I also said he got really sweaty as a result, because it also affected these ones that affect the sweat glands. And then of course, our very first neuron that we learned about, our somatic motor neuron that forms that neuromuscular junction, they also are cholinergic neurons. So with really the small exception of adding this little bit of information here, there is no new information on the board. This is just a summary of stuff we've already talked about. We already know that all these neurons release acetylcholine, and now we've given them a fancy name. All right, questions on that? Here's something else we've talked about as well. Cholinergic receptors. Remember before we had this pesky issue. Is acetylcholine excitatory or inhibitory?
It can be either.
Yeah, it depends. Remember we talked about when we released it onto skeletal muscle, it is excitatory. Whereas when we release acetylcholine on cardiac muscle, it is inhibitory. And do you remember why we said that could occur?
The types of channels.
Exactly. It's because both of these have acetylcholine channels, acetylcholine receptors, but there are 2 different types. There are 2 types of acetylcholine receptors. Now, it would be super easy for us if they named these A and B, but are anatomists going to let us get away with anything simple like that? No. So we have an issue. We need to be able to tell these apart. Let's say we have a third target, and we don't know what it is. It has a receptor, and we'll put this receptor in black. We don't know if it's an A or a B receptor. Now, if we release acetylcholine and acetylcholine binds to it, does that necessarily tell us which it is, whether it's A or B?
No.
No, because acetylcholine binds to both A and B. So using acetylcholine won't help us figure out what this one is. But luckily, acetylcholine isn't the only thing that binds to these receptors. They both bind to receptors, but remember when we talked about the neuromuscular junction, we know toxins, like for instance, curare or tetanus, they can bind to a receptor and have an effect. And it turns out while A and B are very similar, there are different toxins that can affect it. For instance, toxin 1 binds to the A receptor and activates it, whereas toxin 2 binds to the B receptor and activates it. So if I want to know what this unknown one is, instead of releasing acetylcholine, I can release toxin 1. And if toxin 1 binds to it, is this an A or a B receptor? A. If toxin 2 binds to it, is it an A or a B receptor? B. So notice we can use the toxin to figure out which one they are. And so that's how they name them. This first receptor is the toxin 1 receptor. This green one is the toxin 2 receptor. Now, of course, did we get away with terms like toxin 1 and toxin 2? No, of course not. But they use the actual name of the receptors. And it turns out toxin 1 is nicotine. Where do you get nicotine from?
Cigarettes.
Cigarettes, tobacco, technically tobacco, right? Because you can get it from cigarettes, cigars, vaping, all of those things. It's from tobacco. Absolutely. Tobacco is able to bind to this receptor. And it turns out nicotinic receptors, ones that nicotine will bind to, are the ones that are always excitatory. So again, as we talked about, think back to that very first cigarette you had. It made you very alert and twitchy, all right? It constricted, it excited the blood vessels going to your digestive system, decreasing blood flow. So you may have gotten nauseous, may have even thrown up as a result of it. And for some of you, it released an increase of dopamine in your cerebral cortex, especially in the insula, leading to an increased desire to want to do it again, leading to that addiction. All right. So here, oops, I horribly misspelled always. It is always excitatory, and that is that nicotinic receptor. And that's what's found on skeletal muscle. That's what's found in a lot of locations. The second toxin, and again, most of us know what nicotine is. That's something we're pretty familiar with. The second is actually the toxin that was found in those mushrooms I was talking about. And that toxin found in the magic mushroom, not the magic mushrooms, in the poisonous mushrooms, is a toxin called muscarine. I'm going to have to erase my cells now. The second toxin is a toxin found in poisonous mushrooms. It's found in some frogs, poisonous frogs, right? Some people lick frogs to get high. That is the muscarine that they are ingesting when they do that. And so that is the toxin 2 that binds to the second receptor. And its effect is much more complicated. Sometimes it's excitatory, sometimes it's inhibitory, depending on the the organ. So like we saw in the cardiovascular system, it slows the heart rate down. So it decreases the heart rate as a result of that, from that muscarinic receptor that is there. So there you go. We already knew there were different types of receptors, and now we've just given them fancy names based on the toxins that can influence them.
So how do you know though, like for example, for like the skeletal muscle, toxin one's always excitatory, which would be the nicotinic receptor, but
but think about it. When we talked about the neuromuscular junction and our somatic motor neuron released acetylcholine, what was the effect on the skeletal muscle cell?
It would contract.
Well, it would stimulate it to produce an action potential. So it was always excitatory. Remember, we've talked about how our pre-ganglionic neuron always releases acetylcholine, and it is always excitatory on the postsynaptic neuron. Guess what type of receptors are always present on those postganglionic neurons? Nicotinic. So anytime we've said it's always excitatory, this is why. Because it always has that nicotinic, and nicotinic is always excitatory. Other places we said it could be negative, it could be positive. When we said, negative, or it could be positive or negative. When we've said it could be excitatory or inhibitory, then that is typically using the muscarinic. All right. Excellent. So again, not a lot of new information here. We've just given names to things. We already knew that they were because of the different effects on skeletal muscle and cardiac muscle. We already knew there were two types of acetylcholine receptors. Now we've just given them fancy names. And if we're going to give fancy names to the ones that release acetylcholine, then a neuron that releases norepinephrine gets a fancy name as well. Any neuron that releases norepinephrine, we call an adrenergic neuron. And if you notice, we've only identified one of those. Most of the postganglionic sympathetic neurons release norepinephrine. So basically, they're the ones that influence our effectors. So that's super easy. Only one neuron that we've talked about is adrenergic. But here's where things get fun. There are two main types of adrenergic receptors. We can start like we did before by labeling them A and B. But again, are we going to get away with A and B? No. Although we did get close this time. Our anatomist wanted to use A and B, but they of course had to fancy it up a little bit, so they used the Greek. And unfortunately, thanks to COVID, we're learning all about the Greek alphabet. So what are the first two letters in the Greek alphabet?
Is it alpha and beta?
Alpha and beta. So our two main types of adrenergic receptors are alpha and beta. There are two specific alpha receptors, and they are known as, and again, we got to use the Greek. So the Greek alpha looks like a little fish on its side, alpha 1 and alpha 2. And there are three specific betas. Beta looks like bee that's dragging ass, a little bit of a loop hanging down. And they're beta 1, beta 2, and beta 3. So when we look at these three different types, two specific types, and two main types, alpha and beta. And then there are 2 specific alphas and 3 specific betas. So now that I've done that, let's look at it with the pretty words. Alpha 1. Alpha 1 is the most common type of adrenergic receptor. So it's found in most locations. And this is the one that primarily stimulates the blood vessels of smooth muscle. So again, when we stimulate smooth muscle, it constricts. So notice this is constricting the blood flow to the stomach, constricting the blood flow to the mucous membranes of your nose so that you produce less mucus, to your lacrimal gland so that you produce less tears, salivary gland so you're producing less saliva, right? It's constricting all of these blood vessels going to all of these different locations, right? So we have this alpha receptor here. Alpha 2 is inhibitory. And this is the one that decreases metabolic activity. Primarily, like I said, to the digestive system. So you're producing less saliva, producing less mucus, producing less gastric juice. These right here are one of the main reasons why they tell you not to swim until at least 30 minutes after you've eaten. Because if you have a big, huge meal and then you go out to the middle of the lake, as you're vigorously swimming around in your lake, your sympathetic nervous system is decreasing all of the activity of your digestive system. And if you had a very big meal or a very complicated meal or a very fat dense meal, with less secretions and less motility and less activity, you're not going to be able to completely break that down. And if you can't completely break it down, that can lead to intestinal cramping. Right? That's why typically those endurance athletes, right before they go, they have a very simple basic, like, carbohydrates, basic carbohydrates, something that's really easy for the stomach to break down, for the digestive system to break down, right? And again, by pasta, I don't mean that they're having a big, huge dollop of Alfredo sauce on top of it, right? Because with all that fat, it's going to be much more complicated. They're basically very simple, very straightforward meals, or they've got those carbohydrate goops or gels or things like that that they'll eat while they're doing it so they can get that quick burst of glucose, but it doesn't require a lot to break it down. These, like I said, are more common. They're found a lot in the digestive system and stuff like that. The betas are found in other areas. Beta 1 is almost exclusively found in the heart. Its job is to increase the metabolic activity. So when I get up and I'm walking up 3 flights of stairs, or when I'm exercising, or when that bear with an axe walks in the room, my epinephrine and norepinephrine stimulate my heart, and my heart beats stronger and my heart beats faster. And that is a very, very good thing. Unless I just had heart surgery. If I had heart surgery, every time I walk up the stairs to go to my bedroom, do I want my heart beating stronger and faster in response to that if I just finished having heart surgery?
No.
No. So typically after someone's had heart surgery or after someone's had a stroke, or if someone has really high blood pressure, what do we typically give those people? What's the medicine? Beta blockers, right? Most of you have heard the term beta blocker before and you had no idea what the heck that was. Well, now you know what it is. Beta blockers block these receptors. So now when I go up the stairs, my sympathetic nervous system releases that adrenaline to tell my heart to beat stronger and to beat faster. But if those beta blockers are glue in the locks of those channels, is my heart going to beat faster? Is it going to beat stronger as I walk up the stairs? No. And so it helps me to maintain a more stable and a lower heart rate while I'm recovering from that surgery. The second type are the Beta2 receptors. These are mostly found in the respiratory system. And their job is to relax the smooth muscle, open up the airways to get more air inside. Because again, when you're vigorously active, walking up those 3 flights of stairs, exercising, or fighting that bear with an axe, you need oxygen for all of that physical activity. And these two right here are also exactly why I carry an EpiPen anywhere I go with little. As you guys know, my youngest daughter has a severe peanut and tree nut allergy. And if she were to come in contact with the pea protein, the protein from the peanut, it may be possible that she could go into anaphylactic shock. Part of that is a massive vasodilation of the body, which dramatically drops blood pressure. And if your blood pressure drops, is it easy to get blood to your brain? No, but that's okay. Your brain doesn't need blood all the time, does it? No, yeah, absolutely it needs it all the time. And it causes an increase in mucus production. So your airways fill with mucus. So even if you can get blood to your brain, you can't necessarily get the oxygen that you need to get to your brain. A very, very dangerous and deadly combination. So when someone goes into anaphylaxis, what do? Jab them in the leg with the EpiPen. And what does that epinephrine do? It stimulates the heart to beat stronger. It constricts all those blood vessels that we talked about, bringing the blood pressure up, trying to get the blood to the brain, and it relaxes the smooth muscle to try to open up the airway to get the air inside so that she can get her oxygen, she can get it to the brain, and hopefully she can survive. However, as important as all of these are, the one I say these stories over and over again, but again, the world has changed a lot over the past couple of years. 2 years ago, before we were before the world ended with COVID, there was a tremendous amount of money and a tremendous amount of research going into the beta-3 receptors. Because beta-3 receptors are receptors that are found primarily on our adipose, associated with our adipose tissue. And what they do is they help to increase metabolism, they help to increase the temperature of the body, and they help to stimulate lipid breakdown for energy instead of glucose. All right? Why bother going to the gym and working out when you can simply just pop a pill that will stimulate your beta-3 receptors, increasing your metabolism, melting the fat just off of your body, making us all beautiful on the outside? Because after all, as we know, being beautiful on the outside is the only thing that matters. And you could do it the hard way by going to the gym, or if you were able to just sit on your couch and pop a pill and eat Cheetos while you melted that fat away, right? That is a billion-dollar industry. And so a tremendous amount of pharmaceutical companies are looking at ways to take advantage of this beta-3 receptor to see if there is some magic pill that can make them rich and all of us beautiful on the outside. Of course, then the world ended, and I have no idea where the research is on this stuff. But for a while there was a tremendous amount, billions of dollars looking into these beta receptors when they were first discovered because of, again, the potential money-making risk. The combination of we all love beautiful people and we're all incredibly lazy. So if you can put that together in a pill, then that's a win-win, at least for the shareholders of the pharmaceutical companies. All right, questions on that? All right, excellent. Your book's got a great table that talks about these receptors, where they're found and what they do. So take a look at that. The last thing we need to talk about, how are we on time? We're doing perfect. The last thing we need to talk about is, again, adding labels to things we've already talked about. If you remember, when we talked about the neuromuscular junction, where their somatic motor neuron releases that acetylcholine that binds to those receptors on, well, let's make it a shaggy, motor end plate, we talked about two chemicals. One of those chemicals was curare. Remember, curare binds and blocks the receptor, puts gum in the lock so that it didn't function. Whereas tetanus also binds to the receptor. But is tetanus gum in the lock? No, it's a magic key that opens the lock and mimics the effect of the neurotransmitter. So we know that when we talk about toxins, toxins can basically do two things. They can either block a receptor or they can activate a receptor. That is absolutely positively not new information. We talked about that months ago, right? So all we're going to do now is add some fancy names and talk some more examples. An agonist is a toxin that binds and activates the receptor, mimics the effect. One example of this is phenylephrine, which is found in most cold medicines. Remember, alpha-1 was the one that we talked about constricts smooth muscle. So if we constrict smooth muscle to the blood vessels of your mucous membrane of your nose, then as a result of that, less blood goes to your mucous membrane. With less blood, less mucus is produced. And as those blood vessels constrict, the swelling goes down, the mucus goes down, and that big congested sinus that you have from your cold feels better. We already talked about the example of nicotine. Obviously, it affects the nicotinic receptors. And like we said, its big effect is it can affect dopamine, calming you, leading to the addiction. But it also makes you more alert, right? Can affect your digestive function, decrease digestive function, right? Decreases appetite. One of the things that sometimes happen when individuals stop smoking is two things. They're not getting the suppression of the nicotine anymore, so their appetite comes back more robust, and often needing to do something with their hands and their mouth, they tend to eat more just from that as well. So often people, as they stop smoking, can sometimes gain weight because of those. So again, this is not new information. We've just given a fancy name. An agonist is a toxin that binds and activates a receptor. Of course, that means that an antagonist is something that binds and blocks, like curare that we talked about, or my favorite example, atropine, right? You all know my tremendous disdain for chemistry and chemists in general. Nothing good has ever happened from chemistry. Chemistry is stupid. But people who are almost as bad as chemists are optometrists. Optometrists are horrible, horrible people, mostly because they're lazy. All right. They only have eye appointments at noon on sunny days. And then because they're so lazy, when they're looking in your eye, they can't be bothered to tilt their head to the left or to the right to see your retina. So instead, they put these magic drops in your eye, like atropine. And what that atropine does is it blocks the parasympathetic input to your pupil. Remember, as we know, our parasympathetic nervous system constricts the pupil. Well, when we block that, it causes your pupil to dilate. And when they shine that blazing bright light in your eye, can your eye respond by closing the pupil to restrict the light going in? No, absolutely not, right? So they're blinding you with that torch that they carry in your eye. And notice, not only does it block your pupil, but it also blocks the muscles that affect your focus. So after you've gotten those eye drops in your eye from the optometrist, is it easy to read your watch or look at your phone No, you have to hold it way out here to try to get it in focus, right? And like I said, they only do this at noon on sunny days. And then they give you those crappy little plastic wraparound sunglasses that don't do anything. Right? Absolutely, absolutely horrible people, right? And again, and it dries out your mucous membrane, so you get those weird, funky, dry orange boogers as a result of it. Like I said, not quite as bad as chemists, but they're pretty horrible people. And then, of course, another classic antagonist we talked about, of course, those beta blockers. Stopping the heart from beating too strong or too vigorously or too fast when we're recovering from heart surgery or something like that. Again, your book's got a nice table that talks about different agonists and antagonists to just emphasize this point. Again, like I said, the heavy lifting was done. There wasn't too much new information here, just some new vocabulary. Things like cholinergic and adrenergic, things like agonist and antagonist. These are not new concepts. These are concepts we've been talking about for months. We're just adding some fancy names to it. Questions on that? All right. With that, that is everything that we need to know about the autonomic nervous system for this exam. What that is going to leave us with is our sensory stuff. Again, the sensory stuff is 2 days before the exam. So once again, like we did here, it is going to be mostly review, right? After all, we have talked about sensory systems already. We've talked about touch. We've talked about vision. What cranial nerve carries visual information to our brain? Optic. Excellent. Which of our 4 lobes processes visual information?
The occipital.
Yeah. Notice you've already learned about the visual system. Now, again, obviously, have you learned everything about the visual system? No. But most of the stuff we're talking about is going to be review, things like that, things we've already talked about, things we already know. You are only going to be responsible for the material we cover in lecture. And most of it's going to be review, and most of it's going to be fun stuff. We'll do some fun stuff at the end of it as well. All right. There's going to be no major anatomy that you don't already know. Like I said, obviously, we already know about the optic nerve. We already know about the occipital lobe. We already know about the olfactory bulb. So again, there's no new major anatomy that you will be responsible for on this, because just the stuff that we've kind of already covered. We may add a little bit of flavor to it, a little bit of depth, but mostly the major stuff is stuff that we've really already covered. We've covered the tongue and what the 5 tastes are. We've talked about smell. We've talked about vision. We've already talked about a little bit even about hearing and stuff like that. We've kind of talked about these things already a little bit. So it's going to kind of just be an overview. And we also, I guarantee, will have time at the end for an extensive review for both the lab, lecture exam, and the final. So bring your questions. Remember, if you do not ask questions about the material, I assume you've completely mastered the material and I make the exam harder. So come with questions so that we can all be prepared for the exams, both on Wednesday and the following Wednesday as well. This will be our big review day as well, because the sensory stuff isn't going to take long at all. All right. Questions on any of that?
How many questions on the lab lecture for this test do you know, like, about?
I haven't finished it yet. What I would say is that it's not going to be anywhere near as big as the muscle or the bones. I mean, there was so much anatomy in that.
Yeah.
My guess is this will probably be something more in the, I don't know, 60, 65 range, probably somewhere around that. There's not quite as much. There's not nearly as much on this as there was with the bones and the muscles. So yeah, my guess is probably fall somewhere in the 60 to 65 range. Probably a good guess. I haven't finished it yet, so I don't know for certain, but it'll definitely, it's not going to be the 80 questions that we saw on, the muscle or the bones or something like that. No.
And then for the autonomic nervous system anatomy, we have a list of things we need to know for the lab exam. And as for studying for that, are we just looking kind of like, I know we went over it today, the majority of it, but as for studying the anatomy, we're just looking at like the charts and everything we looked at today.
Yeah, textbook illustrations, right? Yeah, exactly. If you think about it, there isn't any histology you need to know for the autonomic nervous system, but let's actually just look at this one, right? Do you need to know that these things here are chain ganglia? Yeah, absolutely, right? So we did, so something simple like that, or really, more like, well, I could do definitely a picture like this, and could I point at this and ask you to know what that is, or that and ask you to know what it is, or that and ask you to know what it is, and so on and so forth. So definitely the anatomy on something like this would be something you could be responsible for. And for that matter, right, where are the octopus pictures? Yeah, something like this, right? Could I ask you, all right, if you, as you mentioned, could I cover up the stuff over here, point at this particular, oops, I got to change the color, point at this particular ganglion and ask you to identify an organ that is innervated by this ganglion?
Yes.
Yeah, so something like that. Or could I ask you to identify this ganglion or identify this ganglion or something like that? Sure, absolutely. Those are the kind of questions that I could ask. Okay. But yeah, I don't think there isn't any histology for the autonomic nervous system, no. So it'll all be charts and models and materials that way. Excellent. Any other questions? All right, last thing I want to remind you about is, again, this is your big study weekend. All right, the exam is Wednesday, 1 week from today. We will get a little new information on Monday, but for the most part, 85% or more of the exam, you now have all the material. So this is your weekend to master that material and be ready and be successful. All right. Questions on any of that? Heck, probably closer to 90. Again, the sensory is not going to be heavily emphasized on this exam. So,
like the eye muscles that we went over, are we responsible for that on the lab?
Yeah, absolutely.
Okay.
Yep.
Okay.
Yep. All righty, excellent. Then you guys have a great weekend. Study hard. As always, I have office hours today. I have office hours tomorrow. I'm available by email, all those things. And then otherwise, I will see you guys on Monday. Have a good weekend.
Thank you. You too.
Thank you.
Bye.